One of the most enduring paradoxes in aging research is the observation that women tend to live longer than men, yet male centenarians often exhibit better physical and cognitive function in extreme old age. This gender disparity in both lifespan and health span is not merely statistical curiosity. It reflects a complex interplay of biology, behavior, and evolutionary dynamics. Understanding why women are more likely to reach extreme old age, yet men who do so tend to age with less morbidity, provides essential insights into the nature of resilience, sex-specific vulnerabilities, and the biological mechanisms of aging. Globally, women outlive men by an average of 5 to 7 years, a difference observed across cultures, socioeconomic contexts, and time periods. This survival advantage is evident from birth, with female infants showing greater resilience to early life stressors and infections. The roots of this disparity are deeply biological. Estrogen, the primary female sex hormone, has multiple protective effects across organ systems. It enhances antioxidant defenses, promotes beneficial lipid profiles, supports immune regulation, and modulates glucose metabolism. Estrogen also appears to protect telomeres, the chromosomal end caps associated with cellular aging, and enhances mitochondrial efficiency, further contributing to longevity. Men, on the other hand, have higher levels of testosterone, which may be linked to increased risk-taking behaviors and higher mortality from accidents or violence earlier in life. Testosterone also affects immune modulation differently and may contribute to higher levels of systemic inflammation over time. This contributes to the observed earlier onset of cardiovascular disease, metabolic dysfunction, and immune senescence in men, partially explaining the sex gap in life expectancy. Yet when we examine functional aging in those who reach 100 or beyond, a subtle but important shift emerges. While women make up roughly 80 to 90% of centenarians, male centenarians tend to be more robust. They show better preserved muscle mass, lower frailty scores, higher cognitive performance, and fewer chronic conditions than their female counterparts. This suggests that men who survive to such advanced ages may represent a uniquely resilient subgroup, biologically and behaviorally. One hypothesis is that male centenarians are more compressed in their morbidity, meaning they accumulate disease and disability closer to the end of life, while many female centenarians may live longer, but with more years of functional impairment. For example, women in extreme old age are more likely to experience osteoporosis, arthritis, and dementia, even if they outlive their male peers. Conversely, male centenarians may have more favorable vascular health and are less likely to be institutionalized or cognitively impaired, indicating a form of survival of the fittest selection where only the healthiest men make it to 100. Genetic studies lend further insight into these sex-based differences. Variants in FOXO3A, APO, and SIRT3 genes are associated with longevity in both sexes, but their expression and interaction with hormones can differ. For instance, FOXO3A, a gene involved in cellular stress resistance and insulin signaling, appears more protective in men than in women in some cohorts. Similarly, mitochondrial DNA haplogroups that influence energy metabolism and oxidative stress responses may exert sex-specific effects due to their maternal inheritance and interaction with nuclear genes modulated by estrogen. Immune aging, or immunosenescence, also follows sex-specific trajectories. Women generally have stronger immune responses, with higher antibody production and more active innate immunity. This advantage helps protect against infection and some cancers, but may also explain the higher prevalence of autoimmune conditions among females. Men, meanwhile, may experience earlier thymic involution, shrinkage of the immune organ-producing T cells, but those who reach centenarian status often exhibit preserved natural killer NK cell activity and lower chronic inflammation than age-matched women. This difference could underlie the lower incidence of age-related frailty and better response to physiological stress observed in long-lived men. Lifestyle factors further complicate the picture. Historically, men have had higher rates of smoking, alcohol consumption, and occupational hazards, all of which increase early life mortality. Women's social networks, health-seeking behaviors, and caregiving roles may enhance emotional resilience and encourage earlier medical intervention. However, men who adopt health-promoting behaviors such as regular exercise, healthy diets, and stress management are more likely to benefit significantly, perhaps due to the higher baseline risk they offset. The growing field of geroscience is beginning to explore how sex hormones, epigenetic modifications, and organ-specific aging differ between men and women. As hormone levels decline with age, particularly after menopause and andropause, the protective effects of estrogen wane, and women's biological advantage narrows. Understanding how to support healthy aging in both sexes may require gender-tailored interventions, ranging from hormone modulation to sex-specific exercise and nutritional strategies. In sum, the gender dynamics of longevity are not about one sex aging better than the other in absolute terms, but about distinct biological and demographic patterns. Women have evolved to survive, often through redundant systems that prioritize resilience over peak physical performance. Men, while biologically more vulnerable in early and middle age, appear to age more successfully when they do reach extreme longevity, possibly due to a concentration of protective traits among survivors. These findings challenge simplistic views of aging and call for more nuanced, sex-specific approaches to healthspan research and clinical practice. Recognizing and embracing these differences can help ensure that interventions designed to promote healthy aging are inclusive, personalized, and effective across the entire human lifespan.